Hi, good afternoon, everyone. Can you hear me and see my screen? Just
We
out of
can.
interest.
Perfect.
Wonderful. Look, thank you so much for the opportunity to present the work that CSRO has been doing. It's led by myself. I'm a team leader within the pest genomics team of CSRO Health and Biosecurity. So before I proceed to say anything, I just want to acknowledge the Department of Foreign Affairs and Trade for funding this work. The work was funded during COVID time, so there wasn't a lot of opportunities to go traveling and collect the samples. So with that, I relied on collaborations from and generosity from across the Pacific and Asian region. So specifically from Sri Lanka, from Malaysia, Fahim, who's in the audience, he helped a lot with collecting Malaysian samples. Indonesia from international coconut communities. From the team there, they provided a lot of Indonesian material. Papua New Guinea as well. And a lot of Pacific Islands collaborators and their generosity has been greatly acknowledged. Also, Ag Research New Zealand provided the historical material which was used to characterize the original CRB-G. So that was really important. Now I'll demonstrate why that's so important to have historical material. Okay, with that, now I'd like to, so very quickly, I think, given that there's so many people within the audience knowing about the CRB and the history of the CRB-G discovery, I'm not going to go into much on this, but this is the seminal paper that was published in 2017, led by Marshall et al. From Ag Research, but also Guam collaborators and a few others. The work originated because Guam had detected around 2006, 2007, detected the presence of coconut rhinoceros beetles. So using the DNA barcoding gene, the CO1 gene, it was discovered that on Guam, the population had a quite unique signature based on the CO1 gene. And from there, bioassay was conducted looking at the efficacies of the nudibranchs and was found that nudibranchs was not effective. And from there, the subsequent survey of the populations across the Pacific identified quite a few locations having similar or identical CO1 signature as the Guam, and hence the legend was born, so to speak. The Guam CRB-G haplotype became a warning and threat to the Pacific region. And this CRB-G really captivated our imagination, especially as scientists and policymakers. And also it impacted a lot of the farmers, growers' livelihood. So you can see that this is just a selection and a very quick survey of what's out there in the World Wide Web. You can see that there's reports on, for example, Solomon Islands, there's IPPC report, there's news from even from the Australia worrying about the arrival of the coconut rhinoceros beetles. Predominantly, it is concerned about the detection and spread of the Guam population, where based on anecdotal evidence and detection evidence suggested that it arrived in Hawaii from Guam around 2013. Not too sure when it arrived in Palau, but Papua New Guinea potentially around 2009, and then subsequently spreading to Solomon Islands and Rota, for example. And again, you have, in Hawaii, for example, in the US, there's really a big considered effort looking at producing videos to educate the Hawaii residents, but also for international community looking at understanding how CRB-G is spreading. Now, in addition to those papers that I just quickly show you, there are also papers, and this is one of the most significant paper by Jonathan Reel and colleagues from University of Hawaii. They looked at whole genome analysis, looking at nuclear genes, around 8,000 single nucleotide polymorphism, and also the CO1 partial gene, because this is how we recognize CRB-G. So this is around 700 base pair, and you can see that the Guam populations are indeed detected from Guam, but also from Oahu, Palau, Taiwan. So multiple locations are having this CRB-G present. Okay, Palau supposedly has two separate populations, as you can see in Palau. There's a cluster that is yellow and another cluster that is purple. What is important from this study, and that's missed by the author themselves, and also missed by the reviewers and subsequent study, is that when we compare individuals that are studied based on whole genome analysis versus partial CO1 analysis, is that based on the nuclear genes itself, Guam actually didn't go anywhere. There is nowhere else in this population study that has the Guam population. However, if you use a CO1 gene, you will see that Guam actually has been detected everywhere. So there's this disagreement between DNA markers that are not recognized by subsequent studies and reviewers and scientists, which is quite disturbing, because this indicate the power of and the attractiveness of using just a partial gene to really track Guam and separate it from other populations. But as a geneticist, we know that DNA markers should agree with each other. So there is a problem here in these studies, and we need to understand what's the actual problems. The next significant population genomic studies came from Australia itself by Kevin Atterbury and group from University of Queensland. Again, involve other collaborators, and they look at again, using the CO1 gene to really detect the presence of CRB-G across the Pacific Island. And here they identify, for example, New Caledonia having Guam populations as well. The disagreement, though, is that CRB-G itself, as we understood it, should be resistant to the nudibranch. However, in CRB-G, some of these individuals, you can see that you can detect the virus itself. So there's disagreement with the analysis of the CRB-G within Solomon Islands and the presence or the claim of its resistant status. The main problem from this study, though, is the fact that the authors assume that the CRB-G markers, the CO1 gene, is the correct marker. And as such, they just included previous studies and just included and then add to it their own population studies, but using the CO1 gene. What is really missing is the historical link to the original Guam population. It is not possible to do a population genetic studies or population genomic studies without including the original question or population that you want to investigate, and then in your conclusion, say something about the original population. So that is a practice that we try to avoid. So why is this important? So a little bit about the mitochondrial DNA genome and the barcoding gene. The mitochondrial DNA genome, as you can see from this slide, is that it is a circular genome. It is inherited from the mother to offspring. So mother will pass it on to daughters and son, and only the daughters will pass on to its subsequent offspring, the same genome that she inherited from her mother. Okay, so there are in total 37 genes, 2 ribosomal RNAs in here that's indicated by the orange genes here, 22 transfer RNAs gene indicated as a pink triangle here. You can see it's dotted across the genome. And then 13 protein coding genes, which are the green ones all across here, of which one of them, this is a CO1 gene, is the one that we're using for DNA barcoding. DNA barcoding is really for species diagnostics, so telling what species are what, okay? By pushing the DNA, the CO1 gene further to better understand, for example, invasion biology, one will need to proceed very carefully. So this is the box region that is approximately 700 base pair that is being used to characterize what species it is and subsequently used to infer the haplotype. So here we have the PCR across this partial gene region, and anything that is in red matched to the original Guam individual that was first reported by Marshall et al. And the assumption here is that because it's inherited as a single genome, here a single circular genome, every individual CRB has the same mitochondrial DNA genome from their mother. If the CO1 is identical here, all the remaining gene regions across every part of here that you can see here, the different genes in yellow, should be identical. However, in reality, that's not going to be the case. So this is a real data here. You can see that Guam, in fact, has two other very unique mutations at two different gene regions, whereas the other populations all have very different mutations across their whole mitochondrial DNA genome. With that in mind, Guam really is very unique. The other populations originally identified as Guam are in fact very different from the real Guam itself. Okay, so that is really important because previous analysis only look at partial gene. Now, if we expand it to the whole genome, which is the whole unit inherited from the mother to the offspring, the Guam is not present in any other population from, for example, from Philippines, Rota, Hawaii, Taiwan, Papua New Guinea, Marshall Islands, or Solomon Islands. One thing that is really important that we can see is when we compare the whole genome, Marshall Island, which is just reported as a new incursion in around October, November last year, match 100% to Solomon Islands. But it is not a Guam because it doesn't have these two other unique mutations. So taking that a step further, when we look at across the whole Pacific and invasive and native range, we can see that Guam is really very unique. However, when we extend the full mitogenome, there's another incursion that is just present in Guam, and that was very recent. There was samples provided in 2022, 23 by the Department of Agriculture of Guam, and we detected that there's a new incursion in Guam. These blue arches indicate that if you were to use partial CO1 gene, all this would have been clustered as Guam. But in fact, when you use a whole full mitogenome, they are not Guam. Furthermore, viruses, when we use the typical PCR markers to detect virus, however, this is based on whole genome analysis, we can see that there is just about a lot of populations with a lot of viruses, including Guam, for example, Samoa, Japan, Rota, Indonesia, Philippines, for example, there are viruses detected as well. So the main question that we really need to ask here now is that if whole genome analysis, in this case, mitochondrial DNA genome, which agrees with the nuclear genome of Rio et al., where Guam did not go anywhere, Guam remained on Guam. Why is it that when other countries, when they were told that they have Guam based on the partial CO1 gene, which is incomplete marker, they started saying that they have the same problem as huge coconut destruction that they can't control with the virus? That is a question we really need to ask, okay? So if we use the mitochondrial DNA genome to push it a step further and reviewing with previous publications, we'll see that we can identify biosecurity hotspots, Malaysia, Philippines, and Taiwan being some of them. And we can identify the pathways. And this is really quite different from the historical pathway that we understood it to be. So this work will be coming up very soon, so keep an eye out for it. It's currently being reviewed by co-authors and collecting the revision to put it into a study. So what are the management options for the CRB-G? Let's just look at historical versus current approaches. Historically, we know that it's the efficacies between entomopathogenic fungi and the virus has been investigated, and it's understood that these two biological control agents has very different mode of dispersal and infection. Furthermore, and this is a really important study by Marshall in 1970 published in Nature, he demonstrated that for metarhizium to work, it needs to work in synergistic effect with the virus. Not only that, the EPF, the fungi, is only at best 30% as effective as the virus. Now compare that with the current approaches. Let's take the case of Solomon Islands as an example. And you can find that from this report that you can download from the internet. Various sections that are highlighted demonstrate how different the approaches has evolved. First of all, conformation of Guam, therefore it cannot be controlled by a virus. Now we know that, at least based on whole full mitogenome, this conformation may be incorrect. So the plan for management then switched to containment and population control, leading to bulk import of the fungus, and then a lot of time spending on refining the artificial breeding site, inoculation methods, minimizing exposure to UV and all that, which are all really relevant, but there is something missing, the Oryctus nudibranchs, that is not being fully explored. Rather, a lot of effort and finances and resources are being investigated into looking at what would be a strain that might be effectively used to control the G, which we know is based on whole genome analysis is not present in Solomon Islands. This demonstrates the importance of getting it right. We wasted a lot of time, resources to potentially let the pests destroy the crop 24/7 for many, many years. Now, just going a bit further, this is unpublished data. We have looked at the virus variation. And what is important to highlight here is that within populations, so these are populations from Malaysia, from India, from Pacific Islands, and so forth, the variation is around 1%, an average of 99.5% identical. However, if you were to survey other region, you will see that you can get up to around 5.5% at least of genome difference. So the question is, will Indonesian, for example, in this case, or perhaps the other isolates in India or in Sri Lanka and Malaysia elsewhere, that might have really big genomic differences that may be used to control the pest. So emerging coconut pests for the Pacific. And we've heard about the red palm weevil, we heard about the coconut, the black-headed coconut caterpillars. I'm going to use the fall armyworm as a really quick example. We were caught unprepared. I'm sure many of you in this meeting, in this webinar, knows about the fall armyworms. Based on the CO1 gene, it is reported it came from Western Africa and spread across to Southeast Asia very quickly from a single direction spread. And this is a natural spread, it's almost unstoppable. However, when we look at whole genome analysis, we can identify hotspots, multi-directional spread, and most importantly, we concluded that this is human-assisted spread to many different locations. And this is a behavior, it can be stopped. Now looking at red palm weevil, I've already showed this slide Philippines, for example, Malaysia, Indonesia, they are all identified as biosecurity hotspot for the coconut rhinoceros beetles. Now we have all these palm weevils that are present in these regions. Are they going to be the next pest being spread around the Pacific? Even in Australia, we are not immune. This was in 2022. Wrinkled forest bilianus, I think, was found in a hardware store in their plant nursery, even though it can come across to the Pacific very quickly. And if you just do a search whether red palm weevil is present in the Pacific, you see that it's reported in Papua New Guinea, Samoa, Solomon Islands, and Vanuatu. But these are the regions, when you read more, the confirmation is a bit unknown. So potentially they're in Papua New Guinea, potentially in Solomon Islands, but we don't really know. But wrinkled forest is in Philippines, it's in Indonesia, it's in Taiwan. So they're literally right next door to the Pacific. They can potentially get there. And perhaps it is time for a genomic approach to better understand the pathways and look for alternative biological control options. Finally, my last slide, that is black-headed caterpillars. Again, as I've shown, the pathway of spread really depends on what type of markers you're using. And we are trying to apply whole genome approach, population genomics. And I think that this is an opportunity for international collaboration to better understand how to confirm where are the native populations. For example, is it Sri Lanka? Because it was reported early in 1800s, for example. And even in Bangladesh, it was already reported in 1960, in Myanmar in 1924. Is this region, is it the original native populations, or is it further? So we do not know. And what are the other potential biocontrol agents? Are there other novel control options such as viruses and RNAi? These can all be answered using population genomics. So with that, I'm just going to thank everyone for your for putting up, and I know I went over time. Thank you.